Thank you very much for the introduction and also for the invitation. In the past, like 10 years, I have been following MPS and all sorts of new technologies. And in the recent, like past 1 year, I have been impressed where it has gone and achieved, thanks to huge effort from NCATS, consortia, NIST, and also other stakeholders. I think it made it possible to come to this stage, and standardization of the methods and the validation standardization, all these things came to this stage. And recently, I have seen some application of MPS data coming into IND submissions. I think that is very exciting. So as usual, this is my personal view. Of course, FDA is a very large place, and each division and each office makes a slightly different take on MPS and other innovative technologies. But I just wanted to give you a broad and general overview of how we take in such new data. Oh, we missed the figure, but hopefully the rest of the slides are okay. So this is supposed to show you the mission and vision of ISTAND. ISTAND was created about 4 or 5 years ago to accept new tools. We had other DDTs, Drug Development Tool Qualification Program, namely biomarker qualification, animal model qualification, and COA qualification. But FDA has been approached with other sorts of DDTs, and we thought that maybe creating the door for accepting such new technology might be helpful for the community, and we have done so. And so today, my talk is mostly focused on qualification, but I just wanted to mention that qualification does not stand alone. We have all sorts of other components coming into play when we accept new data into regulatory review.
Next slides.
Okay. Right. So we have, so major part of our work is to accept new applications through drug regulatory drug approval process, namely IND and NDA. We have pre-IND meetings to review such new approaches independent from submitting IND. You can also submit data through what the process called drug master file, which will allow you to share data from different programs. If you are a tool developer and wanted to share basic information about your technology, you can submit drug master file, and that can be shared through different programs. And of course, we have very strong scientific community consensus done by consortia and NIH and all academic efforts, and they play a huge amount of efforts and value into regulatory review. We always accept and review scientific publications and standard creations through this process. And then finally, a qualification comes in. We also have pre-LOI, pre-letter of intent stage, where we can have a meeting with submitters, requesters, so that we have a common understanding for how you can create quality submission to qualification programs. We also added letter of support a few years ago for the requesters who wish to go through the qualification, but it's not ready for the prime time. You can also have a letter of support so that you can continue your important work. So what does qualification mean? So qualification process was created through 21st century's cure to create very defined stated context of use. That's the foundation. And then once the tools are created, it can have specific interpretations so that you can use the same tool again and again for different programs, because we do publish qualified tools in our website. Sometimes proprietary information needs to be protected. In that case, for the commercial companies, we will hold off such proprietary information. Yet, I think availability of tool might be useful for the other stakeholders. And of course, once it's qualified, such tool and the data can be used for IND and NDA submissions without further review of the technology. That will give you some confidence and transparency into the review process. Specific for the MPS, of course, recognize the importance of reducing the animal use for the during the drug development. And we believe that MPS will play significant role in this space. It's probably unlikely at this stage to completely skip the animal testing, but at least we should be able to reduce the number of animals, especially higher level animals in the drug development process. Many cases, MPS data are used to supplement current paradigm, not to completely replace and eliminate the current standard. However, in the future, if the accumulation of the data supports such things, we might be able to finally replace with better tools than currently exist. Many qualification submissions failed at the letter of intent stage, and the primary reason is the focus on the context of use. The methodologies or tools that pertain to decision-making by pharmaceutical industry or developers, they are outside of FDA's purview. And if such data is not part of IND submissions, or even if it is contained in IND submissions, but doesn't have much weight in the FDA decision-making, probably qualification is not the right path for such MPS or other tools. Another key element is the medical needs. So you need to identify the gaps in the current system, and we need to have some proof that your new approach or technology will overcome such shortcomings in the current system. And you have to prove that your tool is better than currently available data creation. DDD qualification is done through 21st Century Cures Act registration, and it has to go through three-step process. The first step is letter of intent. As I mentioned earlier, you could submit preliminary inquiry through pre-LOI stage. We would engage in either a meeting or a written response during the pre-LOI stage. Then formally, we will start from letter of intent stage. And at this stage, important aspect is the drug development needs and also context of use. I will describe these two things more in detail in the future slides. And second stage is the qualification plan. This is also very important. It is kind of analogous to IND, and qualification plan should contain all the data source, what kind of data will be provided based on the context of use that is agreed upon with FDA review during the LOI stage. And also how you are intending to analyze data so that we can have clinically meaningful interpretation of the data coming off the tool. And then finally, it comes to the full qualification package. This will contain all the data together with the analysis and the results. And once this stage is done, qualified qualification determination will be issued. This is a little bit complicated slide, but I just wanted to make sure that which stage is most appropriate for your MPS development. As you all know, the drug discovery to drug development goes through discovery, preclinical development, especially the clinical development phases. And mostly during the screening phase, you may have lead optimizations or picking the most appropriate drug candidate in this stage. And then during the preclinical stage, you may want to do toxicological studies and other efficacy studies. So far, for the IND submissions, most of the application came to FDA were focused around discovery. This is outside of FDA purview. So when you are creating the context of use, please pay attention to which stage your tool is most suitable. Your tool might be most suitable for discovery phase, and that is perfectly fine. And you can work with the industry to hone in or refine your tool for discovery purpose, especially that many of the MPS tools are becoming more available and cost is reduced so that you can use it for high throughput screening phase. And that is probably one of the strengths in current MPS technologies. Preclinical phase is a little bit of a gray zone. Some of the applications might be more important for regulatory decision-making, and some of them are probably more towards internal decision-making by the industry. In general, replacing the animal test or reducing animal tests using MPS technologies before animal testing to get earlier read for toxicology and pharmacology might be practical use for MPS, and FDA might be interested in those data. During the clinical development, supportive data creation from MPS technology is a big plus, and we are interested in data coming through such channel. This is another very complicated slide, but I just wanted to mention that evidentiary criteria are driven from all the way from need statement, COU, and benefit and risk. So I already mentioned that tailor-made drug development needs are essential to create very tight context of use. And once context of use is created, you may want to consider benefit and the risk of such tool. Sometimes benefit outweighs the risk, and in that case, we can consider that is a good tool. And the balance between benefit and risk are highly dependent on context of use. Some cases like rare disease and other tools might not be available in that space. You can balance the benefit and risk. And all these considerations will go into create evidentiary criteria that will support context of use. The qualification process goes through validations. First, analytical validation, and then secondly, clinical validation. Analytical validation phase is quite straightforward. You would expect that your tool would meet certain standards and reproducibility, and it has to meet regulatory acceptance for reproducibility and expectations. That would also include how you collect the samples, how you handle the samples, and how you characterize your analytical performance based on the needs for clinical side. And clinical validation will collect the data for how you source the data. Is it prospective studies or retrospective studies? And how you analyze and what kind of statistical tools will be used to validate samples of the data. And then of course, you would go through risk-benefit analysis, and importantly, you would need to understand the value or data coming off from this tool would meet the clinical importance. If certain number is created from a tool, does that number or difference in the number make sense in terms of clinical importance? In terms of ISTAND submissions, we launched ISTAND program. It's still a pilot program. We have launched it in 2020, so it's about 4 years old now. And so far, we have received 32 LOI and QP submissions, and we also had many pre-LOI inquiries since the launch. As a pilot program, we stated that we will accept or review about 3 to 4 applications per year. But we have received so many interesting applications that we have reviewed far more than 4 applications per year. Among the 32, approximately 13 belongs to complex in vitro or in silico model submissions, and about half of them are MPS related. The accepted qualification efforts are all published in the database listed here. And we also provide grant support for efforts to progress the DDT development. And the link is provided at the bottom. Unfortunately, we will sunset this program next year, but we have one more chance next year to apply for DDT grants. I just wanted to tell you one recent example of acceptance of LOI for MPS. This is probably not a big surprise for you all, but we accepted Emulate application focused around human liver chip for prediction of DILI, drug-induced liver injury. The context of use is summarized in the middle section. And at the beginning, as many of the applications, Emulate was focused on a very broad approach for DILI. It is understandable that the developers want to use the tool for many different applications, but for qualification point of view, it is difficult to validate the tool for many different applications. And with the talk with Emulate, we were able to focus their effort on prediction based on the comparator. So in this case, they intended to use this tool for predicting DD when you have a comparator. So if you are developing new drugs that have priority concern because of prior experience with a similar compound, had DD risk during clinical development or post-marketing. Obviously, if you have prior concern in similar drugs, we will be paying strong attention to if same risk applies to new drug. And this tool will be able to compare the DD risk between the original comparator and the new drug so that you can determine during the preclinical phase if the risk is higher or lower than comparator. In our database, this submission, redacted version of the submission is posted on our website together with FDA determination letter, which will contain recommendation and criticism of this approach. And just yesterday, we also issued press release through CDER statement. So if you are interested in, please follow the link at the bottom. We also have 4 other ISTAND acceptance at the LOI stage, and these are listed here, 4 of them. And top 2 might be relevant to CIVM. The first one is the tool to estimate the cross-tissue reactivity for biotherapeutics. Most of the cross-reactivity, tissue cross-reactivity assessment are done through animal models. However, of course, animal model has many limitations, and this technology will create expression of more than 5,000 membrane proteins on the cell surface, and you'll be able to not only screen, but identify the off-target proteins. The second one is assessment of toxicity for the leachables for epidural or intrathecal administration. These are very localized toxicity coming off from packaging of the drugs, containers of the drugs, and typically these experiments are done in mice or rats, but again, they have limitations, and this in vitro collection of in vitro assays will be able to overcome these shortcomings. Last 2 are a bit different from CIVM, but these are use of artificial intelligence or machine learning for assessment of anxiety or depression measurement. Second one, the last one is the tool to use AI-based adjudication of clinical events. So in the summary, we are strongly interested in MPS technologies and the data they can provide. And you would need to create COU based on the clinical needs, medical needs, and then you would develop the validation plan based on context of use. And such context of use has to meet the regulatory purpose, and in the future, such data can be incorporated into IND process through either FDA qualification program or direct submission to IND. That will conclude my talk. Thank you very much for your attention.